BY4 mock, June 2015, question 3. And here are some experiments investigating bacterial growth with different levels of oxygen. And these are left for a while, and look at some terms here, oxic and anoxic. So we're basically saying this has got lots of oxygen there, and that doesn't have much oxygen further down the tube. Most people have done okay with here, just spellings really of things. So there's a word that describes bacteria that will only grow in the presence of oxygen, or will only grow in the absence of oxygen, or would grow better in oxygen but could grow in the absence of oxygen. So try and get those spellings right. Part B, some bacteria will not grow at all in the presence of oxygen, and these generate ATP through, cannot generate ATP through aerobic respiration. So using your knowledge of respiratory pathways, how they can still produce ATP. And so this is tell us about anaerobic respiration. So tell us the part of aerobic respiration that anaerobes do as well. So most people have come up with that. And then give us the steps from your notes, anaerobic respiration, and give us as much detail as you can there. Remember, there's 4 marks, so you're not going to get 4 marks just for saying, well, glycolysis happens on its own. You're going to need to come up with other chemicals that are used, and some of the terms, and why, how can they do this even though there's no oxygen there. Think about the job that oxygen does in aerobic respiration, and what alternative is there in anaerobic respiration. But it's all there in your notes. Part C. Some problems here, particularly with reading these numbers on the graph. One thing you can do is just have a look at the scale of the graph and how much is each box worth on this scale. So here from 0 to 10, and there's 5 boxes. So each box is 2 degrees C. So keep that in mind when you are reading these optimum temperatures. In the mark scheme it does give a range, so you can be a little bit off, but most of the problems are from thinking that's 30, 31, 32, 33, 34 and so on, instead of going up in 2s. A suggested environment, well, can we think of an environment where the temperature would be in this range here? So think of an environment on Earth where that temperature will be true. And for these, well, in this range here, try and think of places, or in fact organisms, where that might be true, things to live there. And over here, well, this is pretty hot. Okay, the answer is not a desert, because a desert would be down here somewhere, so it's got to be even hotter. So can you think of any places on Earth where the temperature is going to be right up here, a kind of boiling kettle kind of range? Using your knowledge of cell biology, suggest 2, let's put it in bold there, 2 biological problems which psychrophiles, so these ones here that are in the cold environment, must overcome to carry out cellular processes. So this is a suggestion, so there's a number of possible answers here, but I would go back to AS knowledge and think about what are the problems of being very cold. So even thinking about your coursework last year and the temperature effects on yeast. What are the problems of being very cold? So things happen slowly. So can you think of 2 processes or things that are going to be affected by cold conditions? Or in fact, if it's freezing, can you imagine what might happen to cells when things freeze? Okay, so there's two sensible suggestions there.